Thank you. Thank you very much for the introduction and for the opportunity to share with you all the work that my lab is doing on NCL and developing new therapeutic strategies for this group of disorders. So NCL stands for neuronal ceroid lipofuscinosis, which is also more commonly referred to as Batten disease. And really this is a group of lysosomal diseases that are related and collectively are the most common cause of childhood onset neurodegeneration worldwide, with an overall incidence of approximately 2 to 4 cases per 100,000 live births. There are 13 distinct genetic subtypes, most of which are inherited in an autosomal recessive fashion. The clinical symptoms include new onset of seizures, cognitive and motor decline following a period of normal development, vision loss, and invariably premature death. It really is primarily a neurodegenerative disease, but recently we've learned that peripheral organ systems are also affected in later stages of the disease. The pathological symptoms, as the name describes, include a lysosomal storage material that is very autofluorescent, referred to as ceroid lipofuscin. And this is found throughout the body in cells that are throughout the body, not just in the brain. There is profound neuroinflammation, and neuronal cell loss are also pathological components of the disease. There really are no treatments for any form of NCL, with one exception, which is CLN2 disease. And in that case, there is an FDA-approved enzyme replacement therapy developed for BioMarin. But all of the other 12 genetic subtypes have no treatment available. So my lab at MGH in the Center for Genomic Medicine really has a central aim of making scientific discoveries that will benefit patients and families. And we do this by studying the genetics of the disease in patients, and we take this information to build genetically accurate disease models that will allow us to understand the mechanism from DNA to phenotype. And through this research, we identify targets for therapeutic development. So 2 decades of research into the genetics of the NCLs has really highlighted that there are at least 13 different genetic subtypes. And while individually rare, what this research has really shown us is that there are actually shared genes and pathways with other more common diseases. And a really excellent example of that is the gene encoding progranulin, which when inherited in 2 mutant copies, this causes NCL. When 1 mutant copy is inherited, it actually causes a different neurological disease known as frontotemporal dementia. The 2 most common subtypes of NCL are CLN2 disease and CLN3 disease. I already mentioned CLN2 disease and the enzyme replacement therapy that is available for that. The rest of my talk will be focusing on CLN3 disease, which has an incidence rate of approximately 0.7 cases per 100,000 live births. And it's actually higher in Northern Europe, where a founder mutation has led to a higher carrier frequency. So in CLN3 disease, this is actually one of the more relatively slow progressing forms of the disease, which means there's likely time to intervene post-diagnosis. Disease onset is typically around 5 years of age and begins with vision loss. And following several years, there are additional symptoms develop, including cognitive symptoms, motor symptoms, seizures, and progressive decline over another decade or so of life, leading to end-stage disease, which is typically around in the early 20s. As I said, there's no treatments available, and there are actually very few in clinical development. There is one AAV9-based gene therapy out of Amicus Therapeutics, but this is still in early stages, and we don't really know much about outcome of that trial yet. CLN3, the gene, encodes an endolysosomal multipass transmembrane protein whose function we don't really understand very well yet. Replacement of the gene through gene therapy is a bit challenged by the fact that it is a membrane protein, meaning there's no cross-correction potential. Furthermore, the pathomechanisms of the disease are not fully understood yet. However, there has been really an enormous amount of research in this area that has highlighted a number of pathways as candidates to target for therapies. In particular, calcium homeostasis, autophagy, and endolysosomal trafficking pathways have been highlighted by the work that my lab has done and others in the field have done as well. We've identified that loss of CLN3 function leads to an elevation of lysosomal calcium approximately twofold higher than what's seen in normal lysosomes. And moreover, we've recently found evidence that CLN3 and the main endolysosomal calcium channel encoded by the mCOLN1 gene have genetic and protein-protein interaction. Now, earlier in this session, you've heard about the work that Yulia Grishuk's lab is doing to develop an mCOLN1 gene therapy vector. And given this exciting data that we've identified in our model systems of CLN3 disease, we've decided to team up with Yulia to test whether overexpression or activation of TRPML1 in CLN3 disease has a therapeutic potential using the gene vector that she's developed either alone or in combination with a CLN3 gene therapy vector, given the evidence that CLN3 may modulate TRPML1 activity. So this is a little bit of data supporting the idea that activation of TRPML1 corrects CLN3 deficiency phenotypes. On the top, you can see that overexpression of TRPML1, which is shown in the green cell on the left panel, corrects an abnormal lysosomal enlargement phenotype in CLN3 patient-derived neuronal cells. So the cells highlighted by the stars, you can see on the red channel have very enlarged lysosomal compartments, and this is quantified on the bar graph to the right. When either the TRPML1 protein is overexpressed or CLN3 itself is put back in, there's a complete correction of this abnormal lysosomal enlargement. And similarly, on the bottom, you can see that using small molecule agonists of TRPML1 in an accurate genetic model of disease corrects the lysosomal storage material to normal levels that would be seen in just a normal cultured cell. So with these exciting in vitro data, we've now started to work towards establishing the in vivo preclinical proof of concept data using gene therapy, again, to CLN3 or to mucolipin-1 or mCOLN1, either as single gene therapies or as a combined gene therapy. And of course, the potential is there as well for combination therapy using gene and small molecule approaches. And we're excited to establish this much needed proof of concept data, hoping that this will facilitate the clinical translation of this approach for CLN3 disease patients, but hope that this may also have potential for other forms of NCL or other neurological disorders with shared genes and pathways. Thank you very much.